So next today we have Shuang Qian from King's College London, who is going to be presenting on developing cardiac digital twins at scale.
Thank you. Hello everyone, I'm Shuang. It's my great pleasure to be here to present our research on developing cardiac digital twins at scale. So first is some background. So why do we need cardiac digital twin? So currently the multimodal data has becoming more and more available, and that was including both the image data, which covering the cardiac structure function, and there will also functional data, for example, from the 12 ECGs, and also from the wearables that we are increasingly using recently. And those both modality has been used to link to different health risk factors, as well as predicting clinical outcomes. However, because they are only analyzing the observations, they usually fail to explain the underlying biological mechanisms behind them. So here I'm going to introduce our approach as cardiac digital twins that will be possibly help us to understand what we observe. So it has been spoken by a lot of speakers previously about what is cardiac digital twin. So cardiac digital twin has been constructed by multi-scale knowledge, ranging from ohmic molecule level and to cellular level and all the way to the tissue level, and thus really provide a biophysics and physiological constraint framework that will allow to conduct various tests, which help to guide the therapy and also help guide the clinical decision making. However, it's important to note that there will be huge variation in different populations that vary with diseases, age, ancestry, and gender. So how can we really capture those variability in those different scales and also in different populations, and therefore finally enable to adopt our CDT technology to the general population, still remaining a big technical challenge. And to build a cardiac digital twin, it's usually involve two steps. The first is the anatomical model generation that usually involves to taking the clinical images like MRI or CT images to then make to computational ready meshes. And this step is usually very time consuming and will it could take days or weeks of time for a specialized engineer to build one patient-specific model from scratch. And the other part is the electrophysiological model construction that we usually require the model to capturing the clinical measurements, for example, like the electrograms and like the electrograms. And so to build a electrophysiological personalized model that will usually involves a large number of parameters, and therefore this step would be even more challenging as it will require to perform a lot of computational expensive simulations to fine-tune the model to match to the patient-specific data. And that will usually require high-performance computing. And our aim here is to try to address those challenges, or to some aspect of it, is that we want to try to see whether it's feasible to create the cardiac digital twins at scale. And to achieve this goal, we really need to think about to develop a unified, low-cost and fast CDT construction framework that will include both the anatomical personalization and also the EP personalization. And I'm going to talk about these two parts that we done in the next few minutes. And before doing that, I'm going to introduce our data. So we are using the data from the UK Biobank. As many may know, that the UK Biobank is a large-scale prospective study focusing on participants in their mid and old age. So they have recruited participants since 2006, and they have already recruited 500,000 participants. And they have collected a wide range of data on them. For example, their medical history, physical measurements, and environmental measures, etc. And there are also a growing range of the genetics and the biomarker data has become available. And the recruitment is actually is still ongoing. And by the last time I checked, it already have more than 70,000 heart MRIs along with their 12 ECGs, which is really a significant data set. And among them, there are already like 5,000 of them has been taking the second time repeated MRIs that will be able to conduct longitudinal studies. And this really provides a wealth of data for us to explore in our project. And here just shows our work on anatomical personalization. So we currently made a comprehensive, fully automated biventricular mesh generation pipeline. So this pipeline would take the raw images from the UK biobank, the senior MRIs, and it will be conducting with a UNet-based auto-segmentation, and then the contour would be extracted from it, and then feed into an atlas-based surface mesh generation, and finally, the volumetric mesh will be generated with it, and adding with all the simulation required details, for example, the cardiac fiber architecture and the universal coordinate system. And this whole pipeline has been written in Python, and currently work as a module, each module separately. And this whole pipeline would only take 5 minutes to process one case on a standard desktop. And we have already processed a cohort of 3,464 cases as the initial cohort. And we are going to make this pipeline to be publicly available soon. And as many may know, that the UK biobank is not open source database, and that will require £10,000 to getting access to it. So to really make the wide usage of our work, so we also made a cohort of representative meshes by sex, age, and BMI from all the 50,000 heart MRIs that we collected from the UK biobank. So what we do here is that we categorize all the patients into specific beings of specific sex, BMI, and age. So if there is more than 3 subjects in that specific beings, so we calculate their average point cloud and made their surface mesh, and then build the volumetric mesh from it. And we have and this cohort will also be made public available along with this pipeline quite soon. So after doing the anatomical personalization, the next step is for EP personalization. As mentioned by the previous speaker, there are many challenges to try to calibrate the model to match with the full morphology of the ECGs. So for us, we are really thinking about the efficiency. So we, instead of try to calibrate the model to matching all the ECG morphology, we are only taking one of the specific features, the QRS duration, to calibrate our model. And that could possibly link to less parameters to enable efficient model calibration. And to simulate the QRS duration, so we need to simulate the synapse activation. So we'll use the Fickle-based approach that will involve 20 tissue-level properties, for example, the Fickle locations, the Fickle activation time, and also the heterogeneous myocardial conduction velocities, etc. And then because we are lack of the torso information here, so we place the heart into a reference torso to reconstruct their 12 ECGs. And this approach would involve another 30 ECG electrode placement parameters to be uncertain. And here we are reaching a big problem is that how can we build the personalized CDTs with all those 50 parameters to be unknown? And the approach we take is that we are leveraging the machine learning power that we trained a Gaussian process emulator that allow us to conduct a small amount of simulations, around 300 simulations, that will allow us to map the input to the output, and therefore this GPE can be served as a surrogate model that we can perform a global sensitivity analysis on this. So that will help us to quantify the total effects of each individual inputs on the output, and possibly help us to identify the most important parameter affecting the QRS duration. And here shows our global sensitivity analysis results. So we have taken 10 samples from our patient cohort based on their gender, BMI, and age. And first we do the sensitivity analysis for the QRS onto the first 20 tissue level properties. And we find that the CV is the myocardial conduction velocity is expanding 73% of the variation of the QRS duration. So we take this conduction velocity and adding with the other 30 iatrogenic locations parameters, and we still find the result is the conduction velocity effect is still quite dominating. That it's so we can conclude here that the myocardial conduction velocity is a dominating parameter, expanding the variation of the QRS duration. And given this is the most important parameter affecting the QRS duration, so we can now reduce our parameter space to identify the personalized conduction velocity from the measured QRS duration from the UK biobank. And this whole process is also made fully automated. It will only take 8 minutes per case on standard desktop, and adding up with the previous anatomical personalization step, it will be only take 30 minutes for one personalized conduction velocity, and I think this would be within the clinical timescales. And then we have processed all the 3,400 cases we have, and we can start to answering questions like, are there any changes in the QRS duration? Can be more explained by the tissue level properties like the conduction velocity, or is more depends on the structure information like the heart size. And what we find here is on the first, on the left, we find the larger QRS duration in male is largely extended because of the male is usually have a large size of heart, and we find the variation in the QRS duration in obese and the aging group can be explained by both conduction velocity and the heart size. Then we move on and we perform the phenome-wide association study to try to explore those biomarkers to see how they are correlated with different experimental and phenotypes and exposure that are available from the UK biobank. That will including like the medical conditions, their lifestyles, their mental health factors, etc. And we have find two interesting points. The first is that unlike the QRS duration shown in the blue dots has been correlated with a lot of cardiac structural matrix, the conduction velocity which shown in the red dot is only associated with the left ventricle ejection fraction, and this really suggests its potential to be used in diagnose the heart failure patient. And the second point is that we find it interestingly that the conduction velocity and the QRS duration are both significantly correlated with a lot of mental health related factors. For example, the longest period of depression, and this has been consistent with the previous established relationship about depression and the cardiovascular diseases. And at last, we have also tested that how this biomarker has been correlated with the clinical outcomes by just training a largest regression model, and it has been credited by sex, age, and BMI. And we also find two important points. The first is that reduce the conduction velocity and the increase of the heart size is strongly correlated with a higher risk of conduction disorder like the vascular block. And this result really demonstrates that comparing to all the known observed biomarkers, this tissue level property may be more accurately reveal the underlying biological mechanisms behind them. For example, the fibrotic alternations. And because this property is intrinsically described the physical property of the myocardial and their interconnections. And the second point we find is that this reduced conduction velocity is more strongly related to neurological and mood disorders, and comparing with the QRS duration and the heart volume. And in summary, so to conclude, yes, we can make the cardiac digital twins at scale and moving on to population-based study. And we have made three main contributions here. The first is that we have built a fully automated biventricular mesh generation pipeline to be very efficient. And we also made a large cohort of representative healthy heart that are available for the general scientific community to usage, and they will be all made open source quite soon. And the second part is that we have also built a fast conduction velocity personalization pipeline, and we have found that this cardiac digital twin derived conduction velocity shows a lot of new mechanistic insight onto different populations, and it also shows new functional association with mental health that somehow related with heart-brain connection, which recently becoming a hotter topics. And we also show the conduction velocity shows some predictive power on diseases and may also help in developing the drug. And this is my acknowledgement, and thank you for your attention.
Hi, really impressive work. Thank you very much. So the estimations you have on conduction velocity, how confident are you of them taking into account how many parameters in the model can actually change that?
So we are summarizing all the possible parameters that we have, and we have also do because of time I didn't mention, we indeed do uncertainty quantification on our work onto the 10 representative mesh that we use our personalized conduction velocity and vary all the 50 parameters to see whether how much will that change our conduction velocity, what's the variation of the conduction velocity we have, and we have find it's like a 10% error, so we can believe our conduction velocity is with that confidence interval.
Can I have another one on the depression on the mental health conditions? How much is medication affecting your results? Have you compounding?
Yes, I have looked into the UK biobank and tried to collecting all the information on the medication they have, and we included all the available data there. So there is not a very specific depression-related medicine data matrix related to that. So I'm not sure about this.
I would expect that the brain-heart connection would affect more the T wave rather than the QRS, unless there is a change in heart rate. So I'm a bit confused about that.
So currently, I'm still working on trying to personalize the T wave. So I didn't really look at the T wave's correlation with this depression yet. So, but this is a very interesting point, and I will keep on researching it.
Thank you. Very impressive research. And I have two questions. One is about the details. How do you calculate the longitudinal CV and the transversal CV? You say you calculate the CV from ECG. Is the both have both CV?
Yes, we have the realistic cardiac fiber. So the myocardial conduction velocity I mentioned here is actually longitudinal CV, and I have scaled the transmural conduction velocity by ratio. It's like 0.442 something. So it's like changing along with the longitudinal conduction velocity in the same. So yeah.
Okay. The second question is how to avoid the effect of the Purkinje system?
So what we took is the approach called vascular-based approach, actually from Carly from Gardner's group, that they have tried to simulate a full Purkinje system and also Purkinje system based on these 5 vascular locations, and they have found very similar results on both. And in our approach, we try to vary the location of those fascicles and to try to capture the variation of the Purkinje system as much as we could.
My question is how to, when you want to calculate the series from the ECG data, and how to avoid the Purkinje system effect. Purkinje system trans the electric signal very fast.
So we are incorporating. So after we find the conduction velocity to be the most important one, we fix the fascicles in those models. So in all the hearts, the fascicle locations are all the same. Yeah, and I'll do the CVVT.
Okay, well, thank you. Let's have you speak now.